All right, today we're tackling a really fascinating medical mystery. How does an entire extra chromosome end up causing trouble for one tiny little gland in our neck? It's a genetic puzzle, and, figuring it out is actually teaching us a ton about how our bodies are put together in the first place.
Okay, so here's how we're going to break this down. First, we'll lay out the mystery. Then we're going to follow the clues, just like detectives, starting with the physical tissue and drilling all the way down to the genetic blueprint. After that, we'll check out what's controlling that blueprint, the on-off switches, if you will, and finally, we'll see how it all comes together in this huge ripple effect.
So, let's get right to it. The big question here is, how exactly does having an extra chromosome lead to a problem with the thyroid? What's the connection? This is something scientists have been scratching their heads about for a long time.
Right. So we've known for ages that Down syndrome is caused by a third copy of chromosome 21. Simple enough. But here's the thing. That chromosome has hundreds of genes on it. So how does all that extra genetic noise specifically mess with one particular gland, the thyroid, so consistently?
Well, for a while now, the evidence has been pointing to a problem with development. The thyroid is basically the body's metabolic thermostat, right? And it seems to be off-kilter from day one. In fact, studies have found issues even before birth in fetuses. And that's a huge clue. It tells us the problem starts really early on, probably when the gland is just starting to form.
So to get to the bottom of this, researchers went straight to the source. They looked at the physical evidence, putting thyroid tissue under the microscope, comparing samples from fetuses with Down syndrome to those without.
And wow, check this out. This is our first major clue right here. On the bottom, that's what healthy tissue looks like. See those nice, neat circles? Those are called follicles. They're the little factories that make thyroid hormones. But now, look at the top sample, the one from the fetus with Down syndrome. It's a totally different story. The follicles are smaller, all different sizes. The whole thing just looks disorganized, kind of underdeveloped.
So right away, we have our first solid piece of the puzzle. The thyroid gland itself is physically underdeveloped. The scientists called it lagging maturation, which is just a way of saying its basic structure isn't quite right. The architecture is just off.
But, a physical difference is just a symptom, isn't it? It's not the root cause. So to find that, the investigation had to go way deeper. They had to look past the physical tissue and dive into the genetic instruction manual that was supposed to build it correctly in the first place.
So they used this really cool technique called RNA sequencing. Think of it like reading only the pages of the instruction manual that are currently being used by the cell. And what they found was just wow. It wasn't a couple of mistakes here and there. They found over 1,000 genes that were behaving differently in the Down syndrome tissue. 1,000, that's a huge number. And this graph just lays it all out perfectly. Each of those bars is a chromosome, and the height tells you how much the genes on it are going haywire. And you can see, yeah, there's some noise everywhere, but look at that enormous spike. You can't miss it. That's chromosome 21. It's like the genetic fingerprint of the crime, pointing right back to that one extra chromosome and all the genes on it being overactive. And buried in all that chaos, they spotted something really important. 3 specific genes. Genes that are absolutely critical for a healthy thyroid were all turned down. We're talking about genes that handle everything from the gland's basic development to making and activating hormones. So the key players were being sidelined.
Okay, but here's where it gets even more interesting. It's not just about which genes are being read from the instruction manual. It's also about what's controlling which pages get read. And that brings us to this whole other layer of the mystery, something called epigenetics.
So the easiest way to think about this is to imagine your DNA is a giant cookbook full of recipes. Well, DNA methylation is kind of like putting sticky notes on the pages. Some notes might say, ignore this recipe, while others say, definitely make this one. These little tags don't change the recipe itself, but they control which ones get used.
So when the researchers looked at these epigenetic sticky notes, the results were just crystal clear. This image here is a heat map of those patterns. The top group is the Down syndrome samples, the bottom is the control group. Just look at the difference in color. It's so dramatic, so distinct, you could tell the two groups apart just by looking at these patterns. It's a completely different regulatory landscape.
And it wasn't just a few switches either. They found 266 different spots across the entire genome where these regulatory switches were set wrong. Think about that. This isn't just a couple of typos. This is a widespread, systemic problem with the entire genetic control panel.
Okay, so let's recap the clues. We've got a physically underdeveloped gland. We've got over 1,000 genes acting weirdly, and we've got hundreds of those regulatory on-off switches flipped the wrong way. Now, let's put all those pieces together and see what picture emerges.
And this, right here, is the big reveal. This chart brings it all together. On the bottom, you've got gene activity, how loud a gene is. On the side, you have those broken regulatory switches. And look, up there in the top right, those red dots, those are the smoking guns. Those are the genes where both the activity level is wrong and the control switch is broken. It's the perfect storm.
And this is where we get the real answer. It turns out there isn't one single bad guy here. The extra chromosome 21 isn't like a sniper picking off one specific gene. No, it's more like an earthquake. It sends out a massive ripple effect that shakes up and destabilizes gene activity and regulation across the entire genome. And the thyroid's development just gets caught in that chaos.
So as the researchers put it, what we're seeing here isn't just regular hypothyroidism that happens to show up in someone with Down syndrome. No, this is something totally unique. It's a DS-specific kind of thyroid problem, one that's caused by this massive, system-wide genetic disruption.
So why is all this so important? Well, because once you understand how something is broken, you can start figuring out how to fix it. This discovery could lead to much better, more targeted treatments. And it's a huge lesson for science. For these complex conditions, you can't just zoom in on one gene. You have to zoom out and look at how the entire system is thrown off balance.
And that leaves us with a really powerful thought to end on. If one extra chromosome can create this kind of systemic ripple that affects the thyroid, well, just think about it. How many other complex diseases, things like autoimmune disorders, or even some cancers, how much better could we understand them if we stopped looking for a single broken gene and started looking at the whole, interconnected system instead?